Okay, this is a walkthrough of the simple cloning exercise you were given. And the scenario is that you want to clone out a gene for lipase from this bacteria, Pseudomonas, or no, Propionibacterium acnes. Okay, one thing I want to make sure you guys all realize is all organism, genus, species names must be in italics or underlined. So for instance, E. coli, right? You must have the genus capitalized, and the species is always lowercase, and you always have a period and a space. And then again, they have to be underlined or in italics, like here. So make sure you always write organisms' names correctly. All right, so you want to clone this gene and express it in E. coli. So you've been given this information about the gene. Since this is bacterial DNA, you can just I'm not doing this right. You can use bacterial DNA directly because there's no introns. So you don't have to worry about using mRNA like we will in the next one. So what you're going to do is you want to PCR with primers that include, so that you include the KPN1 and EcoR1 sites. So you do PCR on the bacterial DNA. You don't want to cut bacterial DNA the genomic or the chromosomal because if you cut a whole chromosome, here's a bacterial chromosome with KPN1 and EcoR1, you're probably going to get thousands of fragments. So using PCR is a way to get the specific gene you want away from all of the rest of the DNA in the chromosome. Okay, so that's why we use PCR instead of just cutting the bacterial DNA. But we use the DNA as our gene source because there are no introns, no splicing, no modification that has to happen for this gene to be expressed in E. coli. All right. Okay. So the vector or plasmid that you're going to clone into is named this PCR 2.1, and you can hopefully see that there is a KPN1 site and an EcoR1 site. Is that what we're doing? KPN1 and EcoR1, yeah. So when you cut the plasmid with these two restriction enzymes, you're going to open up the plasmid. It's going to have a KPN1 site and an EcoR1, and there will be a little piece that was between these. Whoops. But it's very small. So what you're going to do is cut your PCR product that's your lipase gene, and cut the plasmid with the EcoR1 and KPN1. And you would run those on a gel to make sure your PCR is the right size. So if you put your PCR sample here, you would see a band representing the 743 base pairs of the gene. And if you run your plasmid that you cut, it should open up, and it's about 3,900 base pairs, and there would be a little teeny fragment that would run off your gel. This is the tiny piece in between the KPN1 and ECOR1 sites. It's so small you honestly would not see it on a gel, but you would see this nice band here showing that you did cut the plasmid. Just on a side note, oops, if you have uncut plasmid, I was trying to change colors on this. This is not something we talked about, but uncut plasmid, which means it's circular, it runs differently than linear, and it usually runs as a couple bands, because some of the circular gets tightly coiled and some of the circular stays more open. That's just how we find it in the DNA. And so they run at slightly different sizes than the true linear. That's more information than you need, but some of you may do this one day, so I just wanted to explain that to you. Okay. So you would have your cut DNA. And you basically add the two cut DNAs together. And you ligate. So you add the ligase enzyme. And so what should be happening is your plasmid, and I usually draw it like this. Here's your lipase gene. So you're hoping they come together, the sticky ends overlap where the KPN1 and ECOR1 sites are, and this is what you want. This is your goal. And so you take that, and this is just in a little test tube, and you take that liquid and you put it into bacteria. Oops, sorry. So now you're going to transform bacteria. And then if we look at this plasmid, we could select with either ampicillin or kanamycin. So it has both of those genes. So you transform bacteria, and let's just say we will plate or grow the bacteria on ampicillin plates. Right? So that's going to give you a plate with what we call colonies. And these are all ampicillin resistant. Now, what can happen sometimes is that you don't cut the plasmid all the way, or sometimes the plasmid can go back together. It shouldn't because of the two different restriction sites, but sometimes it does. And so you need to check each of these colonies and see which one actually has your DNA that you're looking for. So we've talked about the cloning strategy. How would you know which E. coli cells contain the lipase gene? So compared to ones that just have plasmid alone. And it happens, trust me. And so what you can do is you can isolate the DNA from the colonies, from the E. coli. And you could do one of two things. To check for recombinant DNA, you could, one, do PCR with gene-specific primers. So the primers you used. And so if you have, let's just draw it like this, if you have your plasmid plus your gene, right, this is what we want, then when you did PCR, oops, I do. A PCR check, you would get your nice little lipase gene. And if there was no insert, no gene, you would have nothing on your gel, which we really don't like negative results, right? You want to, it's always nicer to see something than to have it be negative and be not sure if you messed anything up. So the other option is, so this is an OR, to do the restriction digest on this isolated DNA with those same KPN1 and EcoR1. Okay, and so if you have your recombinant DNA, you will see your lipase gene size and you will see the size of the plasmid. If you have no insert, when you cut it open, you will just see that size of the plasmid. So two different ways to check to see if the E. coli did take up your recombinant DNA. So lipase plus plasmid is recombinant DNA. Okay, so that answers that question. Then you had a little bit harder question that talked about, where is it? Cloning in this husky gene from a wild dog, but this is a eukaryotic gene because dogs are eukaryotic. So you need to start with mRNA and do reverse transcription. So remember that that is taking the mRNA and making it into a copy of DNA. You always want to work with DNA. Okay. But this is your vector that you're going to clone into. And so here's the promoter. So that's going to drive gene expression. And so therefore you've got to use EcoR1 so that that you're cloning it and so that the promoter promotes the gene expression of the husky gene. So if you look back, your husky gene, there's no EcoR1 sites. So you have to engineer these sites on, and you're going to use PCR to create EcoR1 sites or sequences. When I say sites, I also just mean sequence. At the beginning and end of the gene. And how that works is that when you're designing a primer that's complementary to your sequence here, so you would have a primer that's going to lay down here and tell the DNA polymerase, okay, start copying here, and you're going to have one that says, oops, start copying here at the end of the gene. And then what you do is you just put, as a kind of like a little tail, this EcoR1 site, or sequence. and this is too. So it would be, if we're looking at it here, your sequence would be GATTC, GAATTC. And then what happens during PCR is as you're copying your gene, you'll end up copying the ECOR1 sites into that new double-stranded PCR piece of DNA. And so that you'll have the ECOR1 site on either end of your gene. So it's really cool. That way you can engineer sites onto whatever sequence you're amplifying. All right. So I just wanted to make sure you understood that. These sequences, these sites get incorporated into the final PCR product. So now you have ECOR1, and you've got your BAMH1. Oops. Right. And what you're going to do is, again, cut the PCR product and your plasmid PCM99 with ECOR1. Then you're going to ligate the two cut DNAs together. And you're going to transform into E. coli, your ligation. And then you will select for kanamycin resistance. Right, that's your antibiotic resistance. Okay, so now let's talk about this beta-gal gene. So that's your basic cloning strategy. And we kind of answered some of the other questions. It said, like, it's a eukaryotic, so it has introns, so that's why we start with mRNA to amplify only the exons. We talked about how to add restriction sites on there. So the blue-white screening. So what happens is, here's this vector, and here's what you want to happen. You want your foreign DNA. Nope. Come back. Let's see. You want your foreign DNA to interrupt that LAC-Z gene, that beta-gal gene. And so if it does, you get white colonies. So if LAC-Z is expressed, the colonies will look blue. You can see these blue colonies here. And what you want is white, because that means that your gene inserted in here and disrupted the beta-gal gene. So the sorry, my brain. So the E. coli is not expressing the beta-gal, and so it can't metabolize the X-gal on the plates, and so they appear white. Okay, so that's what blue-white screening is. And this is just showing you, you could get something with no insert, that would be blue, or if it inserted in the wrong place, those would be blue. So you would take these white colonies and screen them. Okay, the white colonies should have your recombinant DNA. The issue with only using one restriction site is that your DNA could go in ECOR1 to ECOR1 with that BAMH1 site, so that's the correct orientation, because you want your gene 5' to 3', or start to stop in the direction of the promoter. If it goes in backwards, and this is really your start of your gene here, it's going to express the gene backwards. It's not going to have a functional husky gene. So the way you can screen for this, well, there's a couple things. So how do you know if it's in the right direction? If you had something functional like I made up, that this is a a fluorescent protein that you could shine UV light on. If you shined UV light on it and you saw the fluorescent protein, so the colonies were glowing purple, I think it was, then you would know you have it in the right orientation, because this would not make a fluorescent protein. It's expressing it backwards. But let's say in real life you usually don't have something like that. So you would do a restriction digest with NOT1 enzyme and BAMH1. If you just cut with EcoR1, you're going to know you have your insert, but you're not going to know if it's in the right or wrong orientation. If you cut with NOT1, you will see that here, this would be a fairly small piece of DNA, because there's not a lot of DNA between. But if you cut with NOT1 to BAM, this would be a much larger piece of DNA. So if you have the correct orientation, I'm just going to make up a number, say it's about 500 base pairs, maybe it's more like 200, I don't know, I didn't give you numbers, I realized. You would have a small band and a larger band that's the rest of the plasmid. So you would have this small band and then this big band. If it's in the incorrect, wrong orientation, you would have a much larger band from between NOT1 and BAMH1. So we'll say maybe it's this big. And then you're going to have another band that's probably not much bigger than the plasmid. Whoa. Almost done. Probably like around here. Okay. So you could screen based on that restriction digest with NOT1 and BAMH1 which plasmid was in the correct orientation. And I believe that covers the cloning. So as you look through the answers and through your answers, please let me know if you have questions still. Thanks.